Okay, it's 8:30. Could we get everybody to head to their seats? So I'd like to welcome everybody to Canola Innovation Day, which is the third and final day of Canola Week. Canola Innovation Day was started several years ago by Wilf Keller, and it was initially called Applying Genomics to Canola Improvement. But as the topics of interest and forward-looking topics of interest grew, we renamed or rebranded the day to be Canola Innovation Day. So today is about exploring emerging ideas and opportunities to innovate and drive future advances in the canola industry. And we have a great list of speakers lined up towards this. Along those lines, I'd like to take this opportunity to thank the Canola Week Organizing Committee, who have all contributed to the development of the agenda for the whole week. And it takes several months and many iterations to bring it all together. But we have such a strong representation of individuals from all across the industry. I'm not going to say all their names because they're listed there. And you've probably seen quite a few of them chairing sessions over the past week. So just want to say a great big thank you to the organizing committee. On that note, I did want to highlight a couple people that have stepped off the committee over the past year. So first off, Isobel Parkin has been an integral part of this committee for several years, and she's no longer an immediate part of the committee. She still has a lot of input in terms of the program because we're still going to her for ideas. But just want to say thanks to Isobel for all her contributions over the past several years. And I was hoping Clint Yerke could stand up if he's in the room. Stand up, Clint. So it was about 3.5, almost 4 years ago that Wilf Keller and Keith Downey were looking for individuals to pass the torch to lead this meeting. And I was really fortunate in that Clint was also interested in co-chairing this meeting. And so he was co-chair with me for a little over 3.5 years and really integral in terms of turning this meeting into what it is today with the important virtual component. So Clint has moved jobs, thus stepped down as co-chair, but I just wanted to give a great big thank you to Clint for all you've done for this meeting. So we do get a lot of inquiries over the year in terms of possible speakers. This meeting, as I said, the committee really puts it together. They'll have ideas and then look for speakers that fit within those ideas. If there are individuals who are interested in presenting, or if there's members of the audience that have ideas for sessions or speakers, we really encourage you to talk to the committee members so that we can try to include those things in the program. Probably spring and early summer are the best times to talk to us because we usually start to finalize the agenda towards the end of the summer. So I'd encourage you all to reach out. So in addition to our excellent list of speakers, we have a lot of great sponsors who have contributed to Canola Week. And so I encourage you to look at the back of your agendas and also the rotating slide deck because we're very grateful for these sponsors who've been able to make this meeting happen. So remember that as we get into the sessions, we have a virtual audience. So it's important that if you're asking questions or answering questions, speakers, that you use the microphone so our virtual audience can hear those. And also remember that we have Slido for questions. So for our virtual audience and for our shy audience members, you can use Slido and you can upvote the questions. We do have 2 mics in the room with mic runners, so you can either go up to one of the podiums or grab somebody's attention if you want to ask a question. For those who are collecting CCA credits, we will show the QR codes on the screen at the end of the day, and you can sign at the registration desk. And then I guess before we get into move on into the program, we wanted to acknowledge that we're on Treaty 6 territory and the homeland of the Métis. So Treaty 6 was signed August 23rd, 1876 at Fort Carleton, which is about 100 kilometers north of Saskatoon. And again on September 9th in 1876 at Fort Pitts, which is about 300 kilometers northwest of Saskatoon. And those Prairie treaties did not end in the 1870s, and we still live with these treaties today. So on Canola Innovation Day, we think about our knowledge and creativity within all the people living across the prairies. And we'd like to think about our challenge, which is to innovate in ways that improve canola productivity and profitability, but also protect the land and the people who depend on it. So with that, I'm going to turn the session over to Kati Navabi, who will be chairing the first session.
Well, good morning, everybody. For those of you who don't know me, my name is Kati. I am a canola breeder, and I work for New Seed Canada as an R&D lead for the North America Canola Program. I'm very excited about the session today because we are going to go a little wild and tap into the treasure box of the canola breeders by looking at the genetic diversity. We have 4 speakers and hopefully ample time for Q&A. Our first speaker is Chris Pires. He's joining us virtually from Colorado. He is he's a professor and head of the Department of Soil and Crop Sciences at Colorado State University. He's a passionate scientist and a great mentor who really likes to engage students in co-creating spaces for asking important questions and having innovative solutions. The title of his talk is going to be Cabbages and Climate Change, Domestication and Feralization of Brassica. All yours, Chris.
Great. Can you hear me okay? Yes. All right, I'm going to about...
Yes, we hear you perfectly.
Thank you. Sorry I couldn't be there in person. I love going to Saskatoon. I've been to Saskatoon, Wuhan, and all the places where canola is important multiple times, but I couldn't make it this year because we're still teaching. So I'm not going to talk a lot about Brassica nidus today directly, but I am going to talk about Brassica oleracea, Brassica rapa, and their wild relatives, and tell a little story about how we found feral plants along the way and how they may be useful. So when I get on an airplane and talk to people about what I do for a living, I often tell people I work on the dogs of the plant world, because everybody knows that a Chihuahua, a Great Dane, or a type of dog, but everybody knows that Cabbage, broccoli, cauliflower, kale, kohlrabi are one species, Brassica oleracea, like a dog. And so I'm going to talk about today in 3 little 5-minute vignettes are feral feature and de novo domestication, and then a little bit about wild and domesticated feral Brassica oleracea, Brassica rapa, and then where we're going in the future with this. So just to start the session off and start the day, and I really enjoyed listening to the talks the last 2 days about how canola has been adapted for abiotic and biotic stress and carbon sequestration and all the other things that are important in agriculture. I just want to start with how important it is that climate change is really our existential crisis for agriculture. So, it's important for in terms of productivity, and it's also important in the fact that we have to feed and clothe the world. So one of the things, though, is climate change is moving fast, but we domesticated most of our crops a really long time ago. So, for example, teosinte, we do a lot of work on this, was domesticated in corn about 10,000 years ago. So how do we think about developing new crops? So now we want crops to do the same thing, but maybe we want to move from annual to perennial crops to do things like carbon sequestration. So one way to do that, and this has been talked about since at least 2022, is maybe you go out in the wild and you find a plant like Panicum turgidum, which is already a drought-adapted wild grass, or you kind of close Stigmina, which is a flood-adapted grass, and you make these into crop plants because they're already adapted.
Sorry, Chris. I'm just going to interrupt quickly. We just can't view your presentation.
You can't hear me?
We cannot see your identification.
Oh, I'm so sorry. Share.
Yeah.
Can you see it now?
Yes, ma'am.
Okay, I'm so sorry. I did the sound check, but not the visual check. Okay, well, here we go. Dogs. Plants. Okay, can you see this? I hope so. All right, no worries. So in terms of native domestication, you can take plants already adapted to their drought or flood climates and potentially turn them into grain crops. Another alternative is instead of just doing traditional breeding, you can use gene editing like CRISPR and take a wild plant, in this case a wild tomato like a Brassica and hirsutum, which is already adapted to a dry climate and doesn't need fertilizer inputs, and change a few key loci to turn a very small green fruit into a large red fruit, which you recognize as a tomato. And this idea was introduced in around 2019 by Fernie and Yan as an alternative route toward making new crops. And I did a quick literature dive over the last few days, and there's been, every year there's a bunch of new papers on this topic from 2021 to 2022. And this has mostly been done in wild tomatoes and wild rice, and it's now being moved into a bunch of different species. So there's probably not a way to get Brassica maplus or canola this way, but there certainly is a lot of ways to do this with the other Brassica crops. So what I've been focusing on with my collaborators at University of Missouri and New York Botanical Garden and University of Florida and other places is how do we tackle some of these Brassica vegetables that are produced in places where there's a lot of drought. So for example, California, where there's about a $7 billion, a huge industry of broccoli. We have kale, Brussels sprouts, cabbage, and there's also Brassica wrapper crops, like broccoli raw and apple cabbage. So one solution to this is to do de novo domestication, where you can take a crop wild relative and then turn it into the vegetable of your choice. So for example, of the 8 wild seed genome species, Brassica hilarionis is one that we've been able to self and actually work in the lab with. And for example, you can imagine, because it already grows in a dry place along the coast of the Mediterranean, that maybe there's just a handful of genes that one can use to turn it into a cabbage or a cauliflower or a broccoli. And one of my grad students, Michael Pires, who's been working in Big Yang's lab at the University of Missouri, has been working on him, but also on this project. And we published a little thought piece in 2022 in Plant Cell Physiology about the prospects of taking wilder feral plants and doing work on them like this. And we're not the only ones, there's also other people who have thought about this as well in the last few years. So another idea is to really use the fact that evolution has already solved a lot of our problems. So if we're trying to think about how we find crops that are adapted to the environments that they're in, why don't we just go out in nature and find them? So using the dog, that's the dog world analogy, if you think of the dog being, or your Brassica as being your domesticated dog, it came from a wild ancestor. But of course, the wolves have a lot of genetic diversity, and you have a bottleneck whenever you do a domestication event to have a domesticated plant or animal. And then sometimes they escape and become feral. And this happens not just with animals, but also with plants. And so I'm going to tell you 2 little stories today where we went looking for things and we found the opposite. So this paper in 2021 that McKinsey Mabry wrote, she was looking at domesticated Brassica oleracea, and we went looking for the wild wolves, and we found it, but then we also found a bunch of feral dogs. And then Alex McCalvey in the same year was starting with feral Brassica rapids and was looking for where they came from, and ended up finding probably what the present day ancestors are. So in terms of thinking about plant breeding for the future, this is a very niche area, but one possibility is to think about, can we go out and find feral plants and then poke them, this was for the title of my talk, to say, hey, you used to be a cabbage, now you're a feral cabbage, can we put you on the rooftop of downtown Denver and have you be a cabbage so we can eat? So these are just some pictures of some plants Feral Brassica oleracea on the left and Feral Brassica rapa on the right, where one can imagine using these as potential starting points or ways to discover genes that might be underlying traits that we care about. And then finally, if we find those plants in the wild, can we then engineer them to be things that we care about? So for example, there are feral carrots, there's feral wheat rice, and there's feral brassicas that we can start with. And so this became, interestingly, a hot enough topic that we organized a conference 3 years ago at the Botany meetings with all these different people from all over the world to talk about feral plants and escaped crops, along with wild relatives. And that led to this paper in 2023 in Plants, People, and Planet. And so here we're just talking about how perhaps we can use these similar plants that are locally adapted to the traits that we care about. They have similar genetic backgrounds to the crops we're working on, and then maybe we can use cutting-edge genomics and CRISPR to do things like precision integration. So now I'm going to just tell two quick little I'll say natural history stories, but I think they're a good start to thinking about the progenitors of Brassica nigrus oleracea. So all of you are very familiar with the triangular view. You have these three diploids of the corners, Brassica rapa, Brassica nigra, and Brassica oleracea, and then the three tetrachords, Brassica nigrus, Brassica jitsi, and Brassica carinata. And there's been a lot of work done on not just the pangenomes of each species, but even the super pangenomes of all of these collectively together, along with other species like Raphanus and other things that the brassicas can breed with. But of course, one thing that's a challenge about these super pangenomes is that you have different fluidity levels in subgenomes. So the first question, our first topic we're going to talk about today is wild brassicas and their crop wild relatives. So if you have plants that are domesticated, then how are they related to the ones that are the closest wild relatives? So in the case of Brassica oleracea, we have these wild relatives that are tolerant to all these different traits like heat and salt. So this is just the results of this first paper that we wrote. So if we ask the question of who's the wolf of the Brussels sprouts, broccoli, and cauliflower, how do we go about doing that? So we have these different 9 candidate species, Brassica microcarpa, Phalaenus, and Solaris, and all these other ones, including potentially wild Brassica oleracea. And there's 3 hypotheses from where they came from when we started. The first one is, of course, kind of like a Dhaliwal hypothesis, that they're from Italy or Sicily, where there's most of the wild species are actually from. The second hypothesis, which was out there from the Eastern Mediterranean, wasn't based on genetic evidence, but mostly literature and also linguistics and archaeology. And the third one, which is that the wild cabbages off of England are actually the ancestors of them. And so we wanted to know, well, who is actually the closest wild ancestor to oleracea, and where is this in our domestication? And so to answer this, we looked at 224 sessions of 14 different oleracea crop types and 9 different species. I don't want to show genetic analysis today, but in this paper there was also lots of archaeology and literary evidence. And the first result from this was that many of the domesticated crops are indeed monophyletic, so kale, romanesco, cauliflower, kohlrabi, and so forth. Some are polyphyletic, so for example, cauliflower actually evolved out of broccoli, so broccoli is paraphyletic. But then most importantly, a lot of the leafy vegetables, the kales and the collards and Savoy cabbages, have been evolving there multiple times or growing up. The second result from this paper was that we resolved the fact that Brassica critica from the Eastern Mediterranean was the closest living relative based on our sampling to the domesticated cultivars of Brassica oleracea. And so, in conclusion for this first little study, taking all the data together, genetics, species distribution modeling, archaeology, and literature, we found that Brassica critica is the closest living relative, and they're from the Eastern Mediterranean. And the things that people in the past thought were wild actually were, for the most part, feral. They had evolved straight out to the field, or more often than not, they actually back crossed to wild relatives. And since this paper in 2021, a bunch of other papers more recently have come up with essentially the same process. So what about Brassica rapa? So Brassica rapa, this project was led by Alex McAlpey at the New York Botanical Garden, along with many of us. We started in the other direction. So in this case, we were starting in the New World and saying, where do these weedy Brassica rapa come from in the Old World? From Spain or from other, and then where are they from? And so Alex collected 189 weedy samples all across North and South America and Europe, and also a lot of cultivars. So if you're not familiar with Brassica rapa, it is extremely diverse. There's Graylos, Rapini, Bok Choy, Kamatsuna, Choisun, Mizuna, Napa cabbage. I mean, we're having another project just on the Brassica rapas in Japan right now. They're extremely diverse, especially on the leafy side. The other thing that we found in this paper we're following up is a lot of these things, these morphotypes are actually not monophyletic, they're not one thing. So for example, turnips have evolved either multiple times or they evolved early on and the things that diverged from them. So the turnips that are found in the Mediterranean versus the turnips that are found in the Middle East versus the turnips that are found in China or Bhutan are separate and have their closest relatives are not each other, but leafy vegetables that occur in their same region. So if you make a phylogeny of these different Brassica rapas, what you find are we think that where they came from is the Caucasus area, and there's medium feral ones there as well. And then we have these clades of East and Southeast Asian crops, and then and then a European crop group, and then there's weedy relatives that have evolved out of this to move to the Americas and across Europe. So if you think about conservation, it's actually pretty important to understand where some of these regional areas are where one can have not only plants that need to be conserved in their habitat, but also where maybe we need to look out for risks of transgenes that could be crossed with them. And then in the case of rapa, when we've gone and done distribution modeling of them, we found that their origin is, we think, as of today, somewhere in this area of stameness, so Pakistan, Afghanistan, Uzbekistan. This seems to be where a lot of the center origin is for Brassica rapa, and of course it's very difficult to collect here, but we're working on that with the support of National Geographic and others. So looking at this all together, Brassica oleracea, we think we have the wild ancestor, extant wild ancestor nailed down in the Eastern Mediterranean, and so this is somewhere we did a lot more exploring in Crete and in that part of Europe. And Brassica rapa, we think that their center of origin is in Central Asia, and so what can this mean for Brassica nimbus in terms of its origins? So there's a lot of people that have been working on this and in the last few years, and one thing that we know is it used to be thought that Brassica nimbus had multiple origins based on plastoloci. That could be true, but at least our data has shown that it's more likely that there's been a single origin, but with lots of back crossing, which is why you get these patterns of admixture that are complicated. And then I would also just point out, there's a few times where we're doing a pangenome of Brassica rapa, and we found that people send in samples and they thought it was rapa, but it's actually nimbus. So we know that there's some leafy nimbuses out there that we have yet to explain. Okay, and then for the last few minutes, I just want to talk about where we're taking this. So the first part of this is back to this idea of the triangle U, and how these different genomes interact and how we can add to them in terms of collections and germplasm and traits. And so I was just looking at this paper yesterday. What we're not doing is, with these large labs in China and elsewhere, they're doing 5,000 Brassica nimbus accessions and looking at pangenomes within one species. What we're doing is finding these kind of evolutionary weirdos and odds and ends of wild plants that nobody's going to go climb a cliff and get them off of Morocco and see how they fit into this and how perhaps those traits could add to the growing body of knowledge that the Brassica community is developing. The other thing is looking at these feral plants. So for example, we have collaborators in Argentina, and they've been tracking these feral Brassica nimbus plants and how they've been crossing with Brassica rapa. And of course you can see at the bottom, if you have basically a Roundup ready weed, you can see how that could be a problem moving forward. And then just thinking about this cartoon, I started with it, but I also kind of wanted to disabuse it. It's not a single line going from wild to domesticated to feral. There's constantly integration and backcrossing and hybridization between your latest elite cultivar and something that may have evolved many thousands of years ago. So looking at this 2019 figure, I think this is helpful. Feralization is not linear. So we like to think about, oh, snap, there's this magic moment, and there's some genes that happen, and then you get a dog or a broccoli. But that's not really true. There's usually standing genetic variation, and then things get selected. But as you're going along from wild to domesticated, there's also constant integration. There's plant breeding and plant improvements. We now have genetic engineering. There's admixture with wild relatives as we have bottlenecks that create these ferals, and there's just a constant movement of genes and traits. So in terms of our feral future, this picture on the left is a picture of Brassica kind of found in some ruins, which I think is kind of emblematic of the things that we are finding as we look at archaeology and plants. We find plants that are no longer being domesticated, but they're still growing there, and they still remember their domesticated past. So we were interested in how we can harness the genetic diversity of not only the wild relatives, but also these feral individuals to withstand pressure environments. And so as we move into the future and explore these more, we'd really like to know if you find plants that you're not sure what they are. We'd like to figure out what they are and see if they might be useful. And in 10 minutes, that's all I have. Thank you.
Thank you very much, Chris, for the excellent presentation. I think we have time for a couple of questions. Are there any questions from the audience or in the Slido? If not, I'm going to ask one question. So in the past few days, I'm sure, Chris, you heard that we talked a lot about public-private partnerships, and I'm wondering how much of these wild or feral species you have actually phenotyped or explored or characterized to understand how they can come useful for commercial breeding programs, for example.
No, good. Thank you for ELYs is practical. So for the whole racing rapids, when I was at Missouri, we did have, you grow everything in quadruplicate in the greenhouse, and when we would harvest the plants, we know leaf 1 would be for glucosinolates, or leaf 2 would be for ionomics, leaf 3 for transcriptomes. So we have a bunch of like co-expression things that we haven't published yet that are associated with this kind of evolutionary background. So we always start with seed, like we rarely take things that it's just people send us leaves. So for the rapizonal races, we've done quite a bit, and their relatives, we've done quite a bit. And that's ongoing work. Yeah.
Excellent.
So I'm not sure which traits are of interest to this group, but for example, glucosinolates, we have like a lot of variation in secondary compounds across these and trade-offs between what environments they grow in and what they put their energy. So I think that could be a potential area.
Definitely. Well, thank you very much for joining us virtually. We really look forward to hosting you in person in Saskatoon one day. Thank you.
Bye-bye.
Bye. Okay, our next speaker is Isobel Parkin from Agriculture and Agri-Food Canada. I know that she doesn't need an introduction for this crowd, but for those of you younger generations here, I think it's important for you to know that she has been an instrumental player in an international project looking at the genetics of oilseed sequencing and mapping canola genome. And this project ended up in a lot of the resources that us breeders use very regularly in our breeding programs. So I thought it's important for me to put that out there. And she's going to talk about exploring the recombination landscape of crops today. Thank you.
Great. Thanks, Katty. Does this? I don't, this timer, you need to turn it on. Oh, okay, there we go. Good, perfect, thank you. So thank you for the introduction. Yes, and thanks for asking me to speak. I don't know whether that was a prize or a penance for my time on the committee, but thanks for inviting me. So I'm going to talk a little bit about some of the work that we've been doing to look at recombination landscape in Brassica napus. And I'll get into it, hopefully. How do I do that? So why are we interested in recombination? So recombination is basically a fundamental process that which drives the exchange of DNA between chromosomes, or the exchange of alleles and different variation. And through that exchange, we basically create all the variation that we now select on. So the things that appear in the field and the things that we choose is derived from different recombination of different alleles in your plants, all reliant on recombination. If you don't also, recombination is a very physical thing. It ensures the pairing of the chromosomes, and with that pairing you have proper segregation, which then creates normal sexual gametes, which leads to the fertility in the crop. So it's another essential process that leads to fertile, and which basically drives yield. And so it's essential for that. And then in plant breeding, it's absolutely essential for allowing the plant breeders to derive stacking of beneficial alleles and also getting rid of deleterious alleles. And having adequate recombination and the ability to recombine different alleles into your crop in a relatively short time increases efficiency of selection. So it's important for all these things. And on the other hand, if you have limited recombination and intense selection, then you can get regions of the genome that are basically inherited together. So this is a cartoon. On the right is a cartoon of half of the genome of Brassica napus. And all I really want to draw your attention to is the bright red triangle, which is on one of the chromosomes of Brassica napus. And this is a region that's constantly inherited in Brassica napus cultivars through selection mostly, but because of this lack of recombination in that region. But that region surrounds the FAE locus, which controls lower russic acid, and so it's consistently selected for. And so we get these regions where we have limited recombination, you lose diversity within your genome. So, and this can be shown in, this is a very old piece of work actually. If you know Andy Sharp, this was work that he was done in his PhD. But on the left, we've got a genetic map which represents the result of pedigree breeding, where basically the breeders were asking, the question was, we have this line which these two lines are very, very similar. We assume they should be the same basically, apart from the only thing that we did was introduce low glucosinolates. But we've reduced the yields, so we have a yield penalty in our line. So what was going on? And so when they looked at the genome, the white bits are basically where it's exactly what they expected. It was exactly the same as the high-yielding cultivar, was the same as the low-yielding cultivar, but the black and the hatch bits came from the low glucosinolate line. But low glucosinolates are basically controlled by only 3 loci, and so these are the black bars. So these 3 black bars were the regions that they wanted to bring in, but they effectively dragged in a lot of other regions of the genome, and these other regions of the genome cores sort of give you a yield penalty in this particular instance. So this is where a case, if you had additional recombination, you could more efficiently select lines that have the trait that you're interested in and get rid of the bits that you don't want. And so that's another reason why recombination is valuable. On the right, I thought this was interesting. So this is a much more modern take on looking at low glucosinolates. And so in this case, they looked at the natural variation, looked at the natural variation, and again, they identified the loci, which is the main one is this one, there's one on C2, but they identified the same loci, and if you pyramid all these loci together, you get low glucosinolates. But they tried to recreate that phenotype using gene editing. They just targeted the causal loci, which were under these QTLs, and targeted it using gene editing. But they created, yes, the low glucosinolate phenotype, but they'd actually reduced the seed size. So, and all the mutations that they generated had low seed size. So there are smaller regions that were being dragged in around these natural loci, which presumably were limiting this effect. And so it's an interesting sort of take on, in this case, natural variation worked better than the modern approach. So factors that affect recombination, that limit recombination within the genome, there are some fundamental things that we can't change, and then some other things that we have some more control over. So there are chromosomal features, so the natural structure of the chromosome. You have the centromeres, which are constrictions in the DNA which control the way the DNA segregates. And they are fundamental features that are in every single chromosome in your genome. And these are very reduced in recombination. You can't change that. You can have local structural variation where different genotypes may vary slightly in the order of the DNA, and you can have inversions that change the DNA structure between two genotypes, and you'll have reduced recombination in those regions. And so we can actually now, with markers, identify those regions. And then there are some other things that are sort of interesting. So Chris was talking a lot about domestication. And they found over time that domesticated crops actually have much higher rates of recombination. So it appears that there's been a sort of selection towards increasing recombination compared to in the wild species. And then there are maternal-paternal effects. So you find that so male and female gametes, the sexual gametes, you get higher recombination in some than the others. And this varies depending on the crop species as to whether it's higher in one or the other. And this is presumably epigenetic factors, so other factors that change the DNA and then also affect how recombination takes place. And then on top of that, environmental factors. So things like stress so heat stress can change the level of recombination, not only the rates, but also the distribution down the chromosome. So this is a very busy slide, and it sort of was a reminder, although Chris helpfully did this. But Brassica napus, as we mostly know, was formed from the fusion of two diploids. And so this is one of our genome sequences. And so along the top here, this line here, this is the A genome, which is the 10 chromosomes of Rapa, and then the C genome, which is the 9 chromosomes of Oleracea. But they're actually very similar to each other, so there's a lot of relationship between these two species, and that's shown by these red ribbons. But the thing I sort of wanted to draw attention to was this. If we just look at one of these chromosomes here, this is B9. This shows gene density, so we get slightly less gene density in the middle, but then we get this is in the middle of the chromosome, we have this histogram shows level of repeats. So these are very short, sometimes longer sequences that are represented multiple times, and these are the centromeric regions. And so this is where you get no recombination. So the top line is actually the genetic map. Sorry, I'm like terrible with this thing. Is the genetic map of the chromosome, and so this is the increasing recombination, sort of accumulative down the chromosome. And in the middle, we have limited markers, but you basically see a plateau where there's no recombination associated with the centromeres. So these are sort of black spots or cold spots in the genome that we can't really do much about. So to study recombination, the problem is that you actually have to generate large segregating populations where you have a lot of different events going on so that you can then have a look at what happened in the previous generations. So we basically have to generate large populations to then work out what happened in the making of those populations and where the recombination events took place. And so one of the tools that we use are these sort of structured multi-parent populations. And there's been some talk about this population already in this meeting. So I think there was talk yesterday about in nitrogen use efficiency, and then there's posters. And so this was generated a number of years ago at Canada, and effectively it's proved to be very useful for a lot of different things. And the way it works is it captures variation through having access to a diverse set of lines. So in this case, we used 50 diverse genotypes, and so this is a phylogeny, which represents sort of some of the molecular variation. Again, this is a PCA that separates out the lines based on molecular variation. But also, Sally Vail had looked at phenotypic variation, and so we used both genetic variation and phenotypic variation to identify 50 diverse lines. So this gave us a lot of ability to capture variation within spring Brassica napus. And then on top of that, we then went through a generation of population. Through crossing to a single line, we had 50 separate populations. So 50 separate populations, and then they went through multiple rounds of recombination effectively in generation of a real population, about approximately 2,500 lines. And so that gave us an excellent resource for not only what I'm going to talk about, but also looking at a wide range of diverse traits. So we genotyped all these 2,500 lines, and then that allowed us to look at, sort of compare recombination rates between each of these different populations. And so this is just a summary based on genetic linkage or genetic maps of each of these populations. Here, I've only got a summary of about 20 populations. We're still really in analysis of a lot of this data. But if you look at the 19, these are the 19 chromosomes, the 10 of A genome and 9 of the C genome. And the box basically represents the variation in the length of each of these chromosomes in the 20 different families. So that represents variation in different levels of recombination in each of the families. And if you look at that in a bit more detail, so this is, oh, I've gone back one. At the top here, we have 2 chromosomes, and they show similar patterns. Again, this is along the bottom here, this is the physical length of the chromosome, and on the y-axis is actually the genetic length. So this is the recombination, cumulative recombination down the chromosome. And you'll see these straight lines where basically there's no recombination taking place around the centromeres, but at either end of the chromosome, this is where all the action's happening, and we're having exchange of DNA at either end of these chromosomes. But what I want to draw your attention to really is that in the 20 populations, we're getting a really diverse variation in the sort of rates of recombination. So across the 20 populations we've looked at, we're seeing quite diverse ranges in levels of recombination. The recombination generally occurs positionally in approximately the same places. So often you'll get similar breakpoints where we've had exchange of DNA, but it's happening definitely at different rates. And so there must be some genetic modifiers, and this is known a little bit from the model plant Arabidopsis, that there are genetic modifiers of recombination that can change, either increase the rates or change, sometimes positionally, the rates. But then the thing with Brassica napus, which is it's sort of a challenge, but also makes it a bit more interesting to work with, is the fact that we don't just have homologous recombination. So, and we see this in these graphs where we look at the recombination rate down the chromosome. So we have these recalcitrant regions with no recombination around the centromeres, but then we also, in some of the populations, we're seeing these lines where in some of these straight lines represent no recombination occurring in areas where we would expect recombination to take place. And so why is that? So this is just, again, it's a cartoon reminder of what napus looks like. And so this is the 10 in red is the A genome, and then the blue is the C genome. And the gray represents the synchrony between these chromosomes. And so they're very similar. And what's happening is we're getting homologous recombination. So instead of having normal genetic exchange between chromosomes, where you basically, you normally just exchange alleles between your sister chromosome, we get exchange between the A and C chromosomes. So now we see recombination that's aberrant. It shouldn't be happening, but it happens, where we get exchange between the A and C of Brassica napus, so the oleracea wrapper Brassica napus, and in that way, we can actually create deletions of different regions of the genome and duplications of those regions of the genome. So this is a natural way that napus has actually generated variation. And we kind of knew this when we did the original genetic maps, but it became very apparent when we sequenced the first genome. And this looks very complicated unless you're used to looking at these. But the main thing to draw attention to is that these red regions represent a duplication of a region in the RAPA genome and a loss of that same region in the C genome. And these regions have been selected for over time by breeding, probably. And in this instance, definitely, because this is the low glucosinolate locus. So this region of the genome, in these are different cultivars of Napus, but in each instance, the same event has been selected for, where you've had this exchange of DNA, and it removed a locus that was controlling glucosinolates, and so this led to one of the low glucosinolate phenotype in Napus. So it's a mechanism for generating variation, and we've now worked out ways of actually studying it. So we utilize not only sequence data to look for exchanges and missing pieces of the two genomes, like a duplication of A and missing C, but we can also use SNP markers. And now we're kind of fortunate with the long read data. So we have long read sequence stabilizers to actually really look at this in a big way. And so we've mapped all these regions within the founder lines of our Napus population. And so this is just 10 of the lines, and it's sort of color coordinated. So when you have yellow, that means we have an excess of the A genome, and the opposite, the green here is kind of an excess of the C genome. And for 10 lines, you can see there's actually quite a lot of activity where we've had, in natural Napus lines, exchange of the two progenitor genomes to generate this variation. And that's and it's very important in the fact that it creates variation. Like this is all of our founder lines, but this is transcriptome data for all of our founder lines for leaf tissue, basically, one single sample, but for each line. But I'm just drawing attention to here, because you can see here these are homologous, so these are homologous chromosomes between the A and C genome, and so there we have the red is high expression of genes, and the blue is basically missing genes from the opposite genome. So effectively, it creates this natural variation of expression loss and expression gain. And then, so if you look at the whole pattern of there's massive, you can see lots of reds and blues. And that is basically the natural variation that's occurring in Napus through these types of events. And it happens all the time. It's an ongoing process that you can capture. There's definitely this genetic variation for it. So some lines are better at controlling it than others. And once they have an event, it's more likely to happen again because it creates more events in the same region. So this was just where we looked at 10 lines and we looked at the level of variation for these types of events. These are cultivated lines, or well, not necessarily cultivated, but they're established Napus lines, and they should be stable. But if you look at progeny from each of these lines, you will see a number of these events. And so the circle just shows like these, the red and blue is basically exchange and loss of DNA in progeny from these lines. And so this is an ongoing process in Napus. So it's a mechanism that you can create variation. But it occurs a lot in resynthesized Napus. So resynthesized Napus is where we take the progenitors, the A and C genome progenitors. So basically kind of what Chris was saying, if you identify some really interesting phenotype from one of these wild species, you can actually recreate Napus in the lab. You can cross the two together, you can double it up, and you can make resynthesized Napus. But it's very unstable. You get a lot of these events going on. And we actually utilize that to allow us to try and understand a bit better how natural Napus controls this pairing and so stops. If this happens a lot and you have too much of this loss and gain of DNA, it can create instability and you reduce fertility, and you'll have aneuploidy, loss of chromosomes, and you'll reduce the yield of your lines. So you obviously want to stabilize it and so prevent and only allow it to happen at a small level. And so in doing that, we basically generated a population between a natural Napus, a resynthesized Napus, and in the progeny, we could identify a genetic control which controls this kind of pairing in natural Napus. And so this allows us, by identifying this locus, which controls about 50% of the variation for this phenotype, we can actually sort of manipulate, in theory, the levels of homologous recombination in Napus and create new additional variation. And so there are other ways of doing this as well. So can we really manipulate recombination in a way that we'd like? And that's the thing that we're sort of interested in now. there has been, in Arabidopsis, there's been a huge amount of work done in looking at meiotic genes that are involved in recombination. And they've identified a number of genes that are basically anti-crossover genes, so they control recombination. And in doing so, you can change these genes and you can actually increase the genetic maps, so basically increase the level of recombination in your progeny by changing these genes, affecting these genes. And they've shown that now that not only in Arabidopsis, but you can do in rice and in pea as well. They manipulated these genes. So there is a way to actually change recombination through genetic modification, and so we're interested in doing that. There's also potentially you could use environmental stimulants or other methods to affect recombination. So basically, the conclusions, we do have a very variable recombination landscape in Napus. There are some genetic modifiers for actual rates of recombination we're trying to get at with some of our data. Our main limitation is that we really need to develop large genetic populations for mapping, and so we really need to get around that. And so finding better methods for measuring recombination would make our life a lot easier. But we know there's a lot of novel variation created not only through homologous but also homeologous recombination, and we'd like to manipulate both of these things. And I guess that's what we'd like to lead to, being able to create new variation within Napus itself purely by manipulating some of the recombination and gaining back some of the regions where we've sort of selected for these large blocks of DNA. So a lot of people are involved in a lot of this work. I work closely with Andrew Sharp and his team at Global Institute for Food Security. And I've had a lot of collaborators along the way that have helped with some of the recombination work. I haven't mentioned them all here, but I've mentioned a few. And then I've been lucky in getting funding from not only the provincial agencies, but also along the way, the companies have been great in helping us, assisting us with developing the NAM population. And I'm just going to quickly do this. Rod Snowden sent me an email this morning, and I'd completely forgotten about this, but there's a meeting in Germany next year in September in Gießen, which is Brassica 2025. Usually, we haven't had it actually since the pandemic, and so everyone's very excited about having it back on board. It's usually a great meeting. Gießen's a fun place to go if you're interested, and it covers the sort of wealth of research in brassicas, and a little bit of other things, a bit of camelina probably, a few other little things. But it's a great meeting, and he also asked me if there's anyone interested in sponsoring the meeting. Then, I can put you in touch. Sorry, I'm a bit over time.
Thank you very much, Isobel. Is there any question? Oh, yeah. Sorry. Are there any questions for Isobel? No. So you showed a lot of the A and C genome comparisons.
Mm-hmm.
Have you looked at the B genome, or will you?
You said maybe. I was going to say you sound threatening. Did I do that? We've looked at the B genome, but you know yourself, you did work on this. Yeah. It's very difficult to cross in the B genome. We have been doing a little bit of that, but I guess we're going back to basics. Oh, sorry. In the sense that, so we have a project now where we're trying to introduce some of the wild genome from like the Oleracea and basically from Oleracea. So we now have, because I didn't mention, but the A genome is much more varied than the C genome. So we're trying to introduce new variation into the C genome from Napus by capturing it from wild Oleracea lines. So we're introducing that, hopefully.
All right.
But not the B genome.
Okay. It's tough.
Yeah.
Oh, thank you, Isobel. This is a little something from the organization.
Donations.
Yeah. Thank you. Okay, so our next speaker comes all the way from Kansas. Mike Stamm is an agronomist and canola breeder in the Department of Agronomy at Kansas State University, and he's the coordinator of the National Winter Canola Variety Trials. The title of his talk is Prairie Prospects, U.S. Great Plains Winter Canola.
Well, first of all, I'd just like to thank the organizers for the invitation to be here today. It's really an honor and a privilege to come to the homeland of canola. And this is actually my second trip to Saskatoon. I was here in July of 2015 for the International Rapeseed Congress. And at that time, everybody was complaining about it being too hot. So I guess it's a bit of a winter wonderland this time around. So I want to give some background on winter canola and provide some information on where actually it's being grown in the Southern Plains. And we'll start with the story of wheat. And wheat acres in Kansas have been on the decline, primarily because of the introduction of corn and soy and cotton into our cropping systems. And yields have relatively stabilized. And this is really the case across the whole Southern Plains. Wheat has generally been on the decline. And the primary reason for that is, if you see there in that red circle in the map of the CropScape data, the area where we've primarily introduced winter canola to has been in continuous wheat for centuries, for a century, literally. Some of these fields where we've introduced winter canola have been in wheat production for 100 years. And so there's this inherent need for a winter break crop such as winter canola. And we've shown in our data, as well as farmers speaking about it themselves, that when you rotate to winter canola, you see a tremendous boost in your wheat yields and your wheat quality that year following planting of winter canola. So that primary wheat belt there in southern Kansas on into Oklahoma is the area that we're targeting, but there are other areas in this region where winter canola could certainly benefit our cropping systems. You're probably familiar with the story of canola in the States. Canola acres are generally driven, of course, by spring canola in the Dakotas, and Minnesota, and Montana. There's been a nice steady increase in both acres and production over the years, over 2 million acres now. Our story in the Southern Great Plains is a bit different, as you can see in graph A here. It's been some ebbs and flows, obviously, with winter canola being more of an alternative crop. I could spend a whole presentation talking about what are the challenges, what are the benefits, what are the reasons why you see this ebb and flow in acres and production in the Southern Plains. But at one point in time, approximately 10 years ago, numerous stakeholders with the U.S. Canola Association were saying that the Southern Great Plains was going to become the #1 region for canola in the United States. And obviously, we didn't get there for a number of reasons, mostly related to markets and variable weather. But the potential is certainly there. So our current challenges and opportunities are that, as you saw in the acreage graph, our winter canola acres have certainly plummeted recently. That's primarily driven by the loss of our market. And so we have a new market now where Schooler, a company, has come in and recommissioned this oilseed processing facility. But we are in an oil-challenged environment, and that's primarily driven by us being still reliant on open pollinated varieties, but then we also have weather and inexperienced growers that go along with low oil content. I would say our biggest challenge is really crop consistency, and this is what I'm going to spend most of my time talking about. You know, farmers really want to grow canola for that yield boost in their other crops, but the consistency is what really frustrates them. Crop rotation is certainly essential, but this time around with winter canola, we're hoping that excuse me, I'm just going to move on to the next slide. So I just wanted to give you some indication of what this crop these challenges that we face. They have a saying in the Southern Great Plains that you're never 2 weeks away from a drought, and so drought is certainly our primary challenge. And as you can see from the pictures here, planting time drought is really a problem where we get limited growth, and then when we get that first cold snap in the fall, we see loss. But then we can also have excessive periods of extreme rainfall. So this bottom map here shows excessive rain in May of 2019. And so this really leads to major challenges when you have primarily OPs being the major varieties that are being grown in the region. And then again, you can see in those two pictures on the right that inconsistency from year in, year out that really plagues the farmers. So really the opportunity here moving forward, I believe, is a transition to hybrids. And we've had hybrids grown in our region for a number of years now, but it hasn't been that consistency that we truly need to make an impact. And in a study that I helped with Bayer CropScience, we looked at all of this hybrid data from around 2003 to 2023. And you can see that we do see a yield delta of about 13% when we compare hybrid yields to OP yields. This was really solidified in my mind that hybrids are the direction that we need to go in that 2019 season, where we had, at this one particular location, over 50 inches of in-season precipitation, where it just caused extreme saturation of our soils. And you can see, though, in that picture where we planted the hybrids next to the OPs in this National Winter Canola Variety Trial, that the hybrids had virtually minimal lodging and over double the yields of the OPs. So in order to address this challenge of introducing hybrids into our region, we started incorporating the Agrua system into hybrid parent lines. We started seed production making test crosses in 2020 using R-line testers from private industry. In the first year we had only one R-line available, but in subsequent years we had two R-lines that we used. And then we take these hybrids then to the field, and we're again kind of in the infancy of our testing program with these hybrid combinations, and we've had some distinctly different weather events that have caused challenges with getting the amount of data that we definitely need. But the future, I think, looks pretty bright for hybrid parent line development, and again, we're primarily focused on the female side at this point in time. But there are a number of A lines here that you can see where we have both positive general combining ability for both grain yield and oil, and overall, most of the K-State derived females exhibit positive general combining abilities for survival. And some lines in particular, like this one, KSMS 052A, show both positive GCA for yield and oil. As I said, this is still really in its infancy, and we do need more testers and more normal testing conditions to get a true feel of how these hybrid parent combinations are performing. However, I think one of the most interesting aspects of this project moving forward is that we are able to introgress the TrueFlex trait into a winter background in collaboration with Bayer CropScience. So I often get the question, well, how far north can we plant winter canola? And so colleagues and I came up with this idea of let's model where our best environments are for winter canola, and let's look at survival in particular. So using this huge repository of national canola variety trial data, we looked at over 200 genotypes and 190 site years where winter survival was rated as a % of fall stand. We looked at historical data for these sites from 1979 to 2019, so we're talking about your min-max temperature, your solar radiation, precipitation, wind speed, etc. And then we defined 39 meteorological variables during what we called the cold period. So that first and last time, the mean daily temperature was below 0 in days after planting. So then for each site, we modeled the proportion of the years in which you would see these high, medium, and low classes of survival. And so you can see that in the map. And intuitively, this makes a lot of sense to me because I've witnessed this in the data that we've collected over the years. But we modeled that as you generally move north of that 40 degrees latitude mark, we typically start to see more challenged environments for winter survival. And this is a trait that is largely driven by the variation that we see in the environment. As you can see, 71% of the variation in the survival data was due to environment. One of the most interesting things to me that came out of this study, though, is that we really identified the factors that caused the most problem with survival. So there were three of these meteorological variables: the number of days with mean temperature between -15°C and -10°, the number of times when mean temperature shifts from negative to positive, and then the mean wind chill. We really hadn't understood which of these environmental factors were the most significant, and so this was something that we were pleased to see. Most of the genotypes that we evaluated were classified as either being what we would call semi-tolerant or semi-susceptible. There are actually 7 that were classified as being tolerant, and the majority of those actually came out of our program, and so we could see that we are definitely having some impact on the improvement of this survivability trait. Other opportunities for enhancing survival. This was a study that was conducted by colleagues of mine at the USDA ARS, looking at candidate genes for overwintering. So this was a combination of a controlled environment study as well as a field study. In the field, there were 9 accessions out of these 600 and so genotypes that were planted that actually survived 2 northern U.S. locations in Minnesota and North Dakota. And of these 9, they were able to identify 235 SNPs clustered across 28 loci and 12 chromosomes. And several of these SNPs that they identified match ones that were previously associated with freezing tolerance in this cold chamber Brassica napus diversity panel that they evaluated. One of these genes in particular, the sensitive to freezing 3 on chromosome A6, is very similar to the SFR2, which was previously identified in that cold chamber study. And a number of these genes are associated with cold acclimation or freezing tolerance. And some of them, of the accessions, obviously we've been using already in our breeding program to continue to enhance this trait. 3 of these accessions they clearly pointed out as having promise for enhancing winter hardiness in the future. There are other beneficial key traits that I think could truly benefit winter canola production in the southern plains. One of those being the semi-dwarfing hybrid trait. So this is taking a dwarf female and crossing it to a normal height male, producing what we would call a semi-dwarf hybrid. And so these semi-dwarfs have this shortening of the internodes that protects the crown at a position closer to the soil surface. This is a problem in the fall where we have very warm conditions and no cold temperatures in order to slow the crop down. And as that crop grows in the fall, some varieties in particular, and especially hybrids, are very susceptible to extending that growing position above the soil surface, as you can see in that top picture. And this is truly a recipe for winter kill. So if we have ways in order that we can protect that crown and keep it close to the soil surface, protecting it from our winter temperatures, then that can truly benefit canola hybrids, I feel, moving forward. There was one other study that I wanted to mention that we've provided germplasm for, and that is this root trait study that the Salk Institute is conducting through their Harvesting Plants initiative. This is through a collaboration with Dr. Wolfgang Busch and his lab, looking at genetic diversity of root traits that could potentially benefit carbon sequestration and climate change moving forward, excuse me. I'm excited about this project and some of the secondary benefits that could potentially come out of it. One of those primarily being drought tolerance. As I had mentioned, drought is a significant issue in our region, And so having a denser, more prominent root mass in the soil could obviously benefit winter canola moving forward. So this is just a diversity diagram of the material that we sent to Salk and some of the other lines that they evaluated. And these plants were grown in pots where they washed the roots and then could take 360-degree images of the root mass. And you can see the diversity in our germplasm compared to West Star, the standard check. So how do we change this narrative about crop consistency with winter canola moving forward? So we're working at trying to identify new winter canola production climates, particularly in the colder north. Of course, we would like to move it north of the border. That's still to be determined. We also are very interested in identifying other areas that are best suited for canola, and we're looking at other traits in addition to winter survival. Oil content in particular is one. I think we'll continue to see a reinvestment in canola variety development in our region with greater integration of these desired traits for our commercial varieties and hybrids. And I think, again, TrueFlex is going to be a game changer for our environment because we have very little experience with herbicide-tolerant hybrids in the region, and the herbicide tolerance is truly what producers want in addition to the other important traits like winter hardiness, pod shatter, et cetera. And this reinvestment, I think, is really being led by the interest now in renewable diesel and sustainable aviation fuel feedstocks and the role that canola can fill there. We need these more resilient varieties that provide the consistent production, whether that's with root traits or carbon store. And then we do need to make this transition from an OP-driven market to a hybrid market. So we can take advantage of all of these traits that are available in our hybrid backgrounds, whether that's hybrid vigor, winter hardiness, vigorous fall and spring growth, pod shatter tolerance, blackleg, lodging tolerance, as I had mentioned in that really saturated soil year of 2019, stable and high oil, semi-dwarf, multi-herbicide resistance. That sounds like a lot to incorporate into our varieties. And I was visiting with a colleague of mine at IRC in 2015 about this, a colleague from Germany, and he said, "Mike, what you're asking is impossible. We can't do this." And I challenged him. I said, "Well, is it really impossible? And what if it was possible? What kind of impact can that have in the Southern Great Plains?" I truly and honestly feel like we haven't had the full complement of what we need in order to be that successful, consistent producer of canola now and in the future. With that, I would like to acknowledge all of my collaborators, funding sources, lots of USDA support for the work that we do, as well as Bayer CropScience, the licensees of our products as well, and all the National Winter Canola Variety Trial collaborators over the years that supported this work with data. So with that, thank you very much.
Well, thank you very much, Mike, for the excellent talk. Are there any questions?
I have one quick one.
Jay, go for it.
Hey, Mike. I was interested in the surprisingly high survivability in the Red River Valley area of Minnesota. So I was just wondering if you, maybe not a lot of site years, or is there something else going on there? I was somewhat encouraged, but I don't want to get too carried away.
Yeah. So the question is, why in the Red River Valley there was such high survivability predicted? It is due to site years. And it's either 100% or it's 0 there. And so that 100% is largely leading that. Yeah, so sorry to get your hopes up.
Do we have any questions online?
We do. So first question from online, could the SNP associated with winter survival in winter canola point towards similar loci in spring canola for frost tolerance?
That's a very good question. I would say potentially yes on that, but I don't know with 100% certainty that that would be the case. That would be something that would be very interesting to investigate.
Thank you very much. Any other questions? Yeah, go for it.
Okay, a couple more online. Could you comment on what the oil content range of the hybrids is, and also tell us about blackleg in winter canola in the U.S.?
Yeah, so the question on oil content, typically in our hybrids, we see 41 to 44% oil year in, year out. And I tell stakeholders all the time that if we just switch to hybrids, we're going to pick up at least 1 to 3 percentage points in oil content based on what I see in the data. We can have high years of 46, 47% oil, but that's fairly rare in the Southern Plains because of the heat that we face during grain filling. And blackleg is our major disease, and most of the avirulence genes revolve around 6 and 7, and RLM 6 and 7 in hybrid backgrounds would certainly lend itself to providing the resistance that we need.
Thank you. One last question.
Okay. The question is, which has gotten 3 votes, what would be your message to Canadian farmers from this research?
What would be my message to Canadian farmers? Thank you. We wouldn't be where we are today without a lot of the knowledge that has been generated through years and years of research that's been conducted in Canada. I mean, our production handbook started with spring canola production practices, and we've since modified those to what we need to produce winter canola. So I would say thank you and keep up the good work.
Thank you very much. I think we better finish on this positive note. Thank you very much. This is a little something for you. Thanks. Okay, to wrap up this session and to witness the deployment of and utilizing of all these wonderful genetic diversity that's available to the canola breeders and also the challenges of it, we are going to have Amandeep Dhaliwal from Bayer CropScience to tell us about the enhancing of genetic diversity in Brassica napus. She works as a canola senior product design scientist at Bayer, and she is based in Saskatoon.
Good morning, everyone. Thank you so much, Katie, for the introduction. My name is Amandeep, and I am living in Saskatoon for the last 2.5 years with my family. And I entered in canola community for the last 2 years. So I started working in Bayer, and first week of my joining in Bayer started from Canola Week. So Judd recommended me to come to this meeting and learn about canola in 3 days. No, it was very helpful. First of all, thank you for giving me this opportunity to come and talk about the efforts that Bayer has been putting in to enhance genetic diversity so that we have a sustainable improvement in the traits that our growers need. Also, thank you to the first 3 speakers. They set up foundation platform for me, so it's very easy for me to go through the slides now. So, plant breeders, they have been using differently diverse resources for the last several decades to improve or develop new cultivars or hybrids that can tackle major problems across the globe, and that includes crop productivity, resilience, adaptation to changing environment, sustainability, and food security. So these sources, they bring in improved agronomics in terms of yield, different sources for abiotic, abiotic stress tolerances, and to improve the nutritional quality, especially in canola, so that we can feed growing population. So here I am highlighting a review that is published by Bayer folks back in 2020, and it reflects the perspective of Bayer on how important it is to have a wide genetic base in crops so that we can improve those crops sustainably for the growers. Also mention some of the challenges when we deal with exotic germplasm or diverse germplasms, how difficult it is to bring them into the cultivated species, and what are the different breeding methodologies that can be used in different crops. So if you have time, please go through this review. So I believe when we deal with the diverse germplasm, the success of crop improvement relies actually in the identification and incorporation of those desirable alleles from different sources. And those sources could be progenitors, wild species, land races, and some of them they are discussed in the talks today morning. And, but my focus will be on how we can utilize diploid progenitors of Brassica napus to bring diversity. So we saw beautiful pictures of evolution of Brassica napus, but my here focus is more on the Brassica napus, that it's a very recent amphi-diploid species that is originated from these two diploid progenitors, and when it's the hybridization happens, very few interspecific events happen between these two progenitors. So these are the two reasons that the diversity is narrow in case of napus. And furthermore, for the last couple of years, the more emphasis on improving the quality, that includes zero russic acid and low glucosinolate. So that put a bottleneck further on the genetic diversity base in napus. So talking about the two progenitors, so the diversity exists within these two progenitors compared to the corresponding two subgenomes in napus, and that could be utilized further to bring that diversity from the progenitors in the napus. Again, I think here I'm summarizing what Isobel has talked today morning, but these are very simple pictures. So on the left, so in this slide overall, I'm comparing two subgenomes. So on the left side in the table, where the two subgenomes, they are compared for different genetic group, but I will be focusing on spring canola. So comparing A and C subgenome, there's a more genetic diversity and more allelic variations available in the A subgenome compared to the C subgenome. And on the right side of the slide, sorry, I can't move my head here. On the right side of the slide, that is a genome-wide decay in LD, linkage disequilibrium. So here, both A and C subgenomes are compared of napus, and there's a sharp decline in case of A subgenome, which indicates that more meiotic recombination happened within this subgenome compared to C subgenome. That means that C subgenome is more conserved. And if we want to further improve or bring in the genetic diversity in napus, so we need to focus more on C subgenome. Moreover, many studies have been done in the past which were focused on A subgenome, and one reason could be because it's easy to develop interspecific hybrids crosses between A subgenome and napus compared to, say, C subgenome. And I think when the interspecific crosses, they are developed between napus and oleracea, that needs very sophisticated cell and tissue culture technologies. So for the next few slides, my focus will be on looking into the Brassica oleracea as a source for variation. So this is one of the very beautiful, unique study done by Dr. Habib Rahman's lab. Here, different C genome donors of Brassica oleracea are used together, cabbage, cauliflower, broccoli, and kale. And they were crossed with the common recipient parent, Brassica, in breadline. These seed genome donors, they are diverse morphologically. We saw in the talks earlier also, but they are distinct genetically also. And that's our purpose, to bring diversity not from a single seed genome donor, but from the multiple seed genome donors together. And in this study, the different traits were analyzed, but I will be highlighting 3 main traits. Just to give some background about these graphs, on the x-axis, the red ones, they are different inbred populations that are developed by using seed genome donors of Brassica oleracea with the Brassica napus inbred line as a recipient line. And the green color indicates 2 breeding strategies that are used, F2 derived and BC1, F1 derived. And on y-axis, these are the values for different traits. So comparing within the seed yield compared to the Brassica napus parent, we see variation on both positive and negative side of this trait. For sure, there are some germplasms coming from broccoli, which have a higher seed yield compared to the diploid species, and compared to the inbred line. Similarly, for days to flowering, we do see variation. For example, in broccoli, kale, they are early flowering than the napus parent. And similarly for seed percent, which is at the bottom, the variation do exist. So in summary, so what I'm trying to emphasize on this slide is that the variation do exist in Brassica oleracea seed genome donors, and it will be very beneficial if these seed genome donors, they are utilized together. Again, using one of the population interspecific cross that I showed on the last slide. This is between napus and Chinese kale. Kale is relatively early in flowering than the other seed genome donors. And in this DH mapping population, the 2 major QTLs were identified that came from Brassica oleracea. So want to mention one more point, that whatever the variation we noticed on the previous slide, that variation was originated from Brassica oleracea seed genome. So early flowering QTL identified on C1 and high oil QTL on C5. And on these genetic maps on the right side of each map, we do see those black bar like boxes. They indicates the presence of the same QTL in different environments and in different years, which indicates its stability. So these are some of the examples where the QTLs, the variation do exist for the traits on the favorable side for these seed genome donors. Talking about the most important trait, seed yield. Again, from these populations which are developed between Brassica napus and Brassica oleracea. So here I will be comparing the seed yield in inbreds, hybrids, mid-parent heterosis in terms of the genetic distance. So sorry, this is being some populated slide, but I will walk you through it. On the left side in the graph, it's a seed yield on the x-axis and the hybrids or lines on the y-axis. And we do see a bimodal distribution, but this bimodal is coming from 2 different type of populations. One is inbred on the left side, which are low yielder compared to their test hybrids. In the middle of the slide, on the x-axis, it is the genetic distance. It's the genetic distance from the Brassica napus line, which was the recipient line. So as on the top one, as the distance increases from the Brassica napus line, the seed yield decreased in inbreds. But this relation was positive in case of hybrids. And similarly, the mid-parent heterosis increased as the genetic distance increased from the Brassica napus line. On the right side of the slide, it's the comparison of the heterosis compared to spring by oleracea has almost double the heterosis. So overall point from this slide is that as genetic distance increases, there's a positive correlation for the seed yield in hybrids and mid-parent heterosis. So based on this information, plus we have been talking a lot about a collaboration between public universities and private companies. So here is one of the example where the collaboration is established between Bayer CropScience and University of Alberta canola program, and from University of Canola, it's a Dr. Habib's Rahman lab who is collaborating with Bayer CropScience. So to take this knowledge further to advanced level, where we can deeply dig into the benefit of Brassica oleracea alleles in the background of Brassica napus and how we can take that diversity to the farmers level. Some of the lines which are developed by Habib's lab was coming from Brassica napus and oleracea with extensive breeding selection and with the confirmation of the Brassica oleracea alleles in the background. So some of those lines, they are selected for this collaboration. And now we know that when we deal with any wild progenitors or any exotic germplasm, we do expect some genetic background coming from those donors, as well as could be some linkage track. So we know those issues could come. So that's why efforts were done by Dr. Habib's Rahman lab already, where they focused a lot on the initial stages in the breeding on napus type quality, plus a spring type growth habit. And now we are doing the field testing, extensive field testing over years, over location, so that we identify lines that are promising without any linkage track or without carrying major genetic background from the Brassica oleracea donors. So after field testing, the hybrids will be developed for these lines to study its heterosis, as well as mid-parent heterosis. And then once those are now we have those Brassica oleracea alleles in the background of Brassica napus, then how those traits, they are inherited from generation to generation. And here I put some pictures. So these news, they came early this year that highlights the importance of this collaboration, where these this profitability that can arise by using oleracea with the help of Bayer CropScience, those Brassica oleracea beneficial alleles, they can increase the sustainability, or they can improve the traits in a sustainable way. This collaboration is at its initial, I would say, in the early phases. So just want to mention this is all inbred data, not hybrid. And we know that Brassica oleracea lines, they are vegetative lines, so we do expect some delayness in the flowering, but with extensive selection in the early pedigree selection, plus now with extensive field testing, we are able to retrieve the lines which is flowering same as that of the commercial checks. So on these three graphs, blue indicates the germplasm that we are testing now, and the red indicates the commercial checks. So here inbred germplasm is compared with the hybrid checks. There's one picture, it is depicting the variation in the flowering, and on the left side, that is in the form of the numbers. Even though most of the germplasm, it is on the right side of the average of the days to flower, but the message that I want to convey here is that still that flowering, it overlaps with the days to flower of the commercial checks. On the bottom, there are two graphs. The left one is oil and the right one is the protein. Again, 2 points here. Looking at the width of the variation is huge that we can tap into. And the second, the selection has been done on the positive side of the trait. So we do see a lot of germplasm on the right side of the average, and they are even better performing than the checks. But here, it's a comparison of, again, inbred lines with hybrids. So summarizing my talk, plus this collaboration, so one of the goal of this collaboration is to develop breeders for the future and to connect academia with the industry. There's a huge focus on the talent development. New graduate students, they are being hired and will be hired more under this program that will get exposure to commercial breeding programs, the extent at which field testing is done, data collection, data analysis. So I believe this is a great opportunity for the new graduate students to get first-hand exposure to the commercial breeding programs. Also, another major outcome that we expect is to develop scientific knowledge that will be shared with the canola community and in the form of publications, and that will be that what is the effect of these Brassica oleracea in the background of Brassica napus. And for sure, Bayer CropScience will utilize this scientific knowledge to further improve our breeding programs internally. The lines that will be identified for best heterosis and for best combining ability, that will be utilized by Bayer CropScience to develop better hybrids in the future. So with this, I want to say huge thanks to NSERC that made this collaboration happen between private and public entities. Huge thanks to Dr. Rahman from University of Alberta for leading this project from UFA. Also thanks to Bariso, who is dealing with all the logistics, experiment setup. State is the person who actually put seed in the soil and help us to collect the data. And Anik is the new graduate student who is hired under this program. From Bayer CropScience, huge thanks to Jed, and I'm not able to locate him here. So thank you so much for providing your scientific expertise, for your vision on this collaboration. Thanks to Emily. She was here with us till yesterday, but today she left to Lethbridge. And Deidre and Oscar, they are more involved in the data analysis. So with this, thank you so much for your attention. And yeah, any question, please let me know.
Thank you very much, Amandeep. That was an excellent example of a public-private partnership that we keep on talking about. I guess there is one question on the online and one on the floor. Geminis, you go first.
Okay, thank you, Amandeep, for your excellent presentation. What is your next plan with the two major QTLs that identified on C1 for early flowering and C5 for high oil?
Yes, so that's already a published data, and that's in the public domain. And what I'm trying to highlight is that that is the variation or that is available in the seed genome donor that could be utilized. And we are looking from Bayer side, we are looking for the products that we can take further in the hybrid development program. And Dr. Habib's lab will be looking more on the QTLs that we can use. It's not only for oil or flowering, but it could be for other traits as well. So we are talking about other traits in the morning session that have a huge impact because of narrow genetic diversity. So there's a huge opportunity for us to look for different traits and for their QTLs.
I mean in terms of cloning or developing marker for these traits?
For sure. So for those two QTLs, I believe they have identified for the fine mapping from those regions, they identified related to flowering with a comparative analysis with other crops. So some of the genes related to flowering, they were identified, and they were doing the further fine mapping of those traits. But the fine mapping is not done yet. But for sure, if we want to transfer that QTL or that gene, we need those reliable markers.
Okay, thank you very much. I think we ran out of time, but thank you. And welcome to the canola community, Amandeep. Thank you very much to all of you, to all the speakers and all the sponsors. We wrap this session up, and there's coffee and chocolates on the first row. So enjoy your time. Thanks.